All right, so good afternoon, everyone, and thank you for tuning in for this talk. I'm Roberta, an instructor in the Department of Radiology at the Martinez Center for Biomedical Imaging and MGH here in Boston. And I will talk today about my research on investigating and modulating the autonomic nervous system in humans with the aid of high and ultra-high field MRI. So to give you some context for my talk, I will start by saying that I work in a neuroimaging lab where we mainly focus on brain responsive to aversive symptomatology, such as chronic pain, for example. However, I've always had a personal interest in brain-body interactions, and I have looked for opportunities to explore this topic throughout my career. Over the last few years with my mentors and collaborators, I started focusing on the vagus nerve, since this provides a physical connection between the brain and numerous organs. This cranial nerve carries preganglionic fibers involved in the autonomic regulation of several organs, including the heart and parts of the gastrointestinal tract. And it is responsible for tasks such as heart rate, GI peristalsis, sweating, and other functions. So therefore, accessing the vagus nerve and somehow modulating its activity could constitute a powerful tool for many clinical applications. For example, an established treatment targeting the vagus nerve exists and is used for resistant epilepsy or major depression. It is actually FDA approved. Traditionally, vagus nerve stimulation, or VNS, requires the implantation of a neurostimulating device connected to an electrode located along the cervical branch of the vagus nerve. So, as mentioned before, the vagus nerve is known to be involved in the regulation of heart rate, cardiac contractility, and a whole bunch of cardiovascular outcomes. So VNS could be also promising for cardiological patients. In fact, recent clinical trials evaluating the use of VNS in patients with heart failure demonstrated a significant increase of parasympathetic tone as measured by heart rate variability, and improvement of cardiac function as well. Despite the reported beneficial effects of VNS, its invasiveness and complications, because it's a surgical procedure, have limited the conduction of clinical trials evaluating its effects on cardiac autonomic modulation for other indications than heart failure. Therefore, the development and optimization of alternative non-invasive techniques for vagal modulation is of critical importance. A possible non-invasive option is given by the auricular branch of the vagus nerve, or ABVN, which is known from cadaver studies to innervate the human auricle, so the external part of the human ear, and most consistently a specific portion called the cymba concha. Here. From there, electrical impulses are able to stimulate the vagus nerve transcutaneously. Hence the term transcutaneous auricular vagus nerve stimulation, or TAVNS. Stimulation of the ABVN is primarily afferent, and a number of studies have combined this neurostimulation paradigm with functional magnetic resonance imaging, or fMRI, in order to evaluate the brain response to TAVNS. Even though this has been linked with activation of the lower portion of the brainstem, called the medulla, some technical challenges, and more importantly, a wide range of possible stimulation parameters and paradigms make it more difficult to combine and summarize the results. So in our work, we attempt to optimize TAVNS parameters using brainstem responses from fMRI as outcomes. So as a starting point, we need to know where exactly to look at within the brainstem. The vagus nerve includes axons which converge onto two main sensory nuclei. The nucleus tractus solitarius, or NTS, which receives visceral and taste afferent information, and the spinal trigeminal nucleus, which receives sensory information from the outer ear, the dura, and the posterior cranial fossa, and the mucosa of the larynx. As for the efferent side, the two main motor nuclei are the nucleus ambiguus, which mainly sends efferents to the heart, and the dorsal motor nucleus of the vagus, which sends parasympathetic output to the viscera. All these four nuclei are located in close proximity in the medullary portion of the brainstem. So we have established that we need to interrogate the brainstem. And so at this point, let's consider the structure for a minute. The brainstem connects the forebrain to the spinal cord and acts as a relay and processing station between these parts of the central nervous system. However, it has some anatomical and physiological characteristics that present challenges to neuroimaging analysis. A significant challenge in using fMRI to assess the response of TAVNS in target nuclei in the brainstem is posed by the structure of interest itself. So the brainstem has a small volume relative to the side of the brain. And in addition to the nuclei involved in autonomic regulation, which are highlighted in the red box in this illustration, is involved in the modulation of many other functions and hosts key source nuclei for several neurotransmitters. For example, the RAFI nuclei regulating serotonin release. The reduced size and close proximity of these nuclei, as well as the location of the brainstem, which makes the structure particularly susceptible to physiological noise, calls for specialized imaging methods. So for example, let's consider the periaqueductal gray in the midbrain. If we use 3 mm voxels, it would be covered by roughly 4 voxels. So something like this. However, this nucleus is involved in a variety of functions with functionally distinct subportions that receive inputs and project to different structures. So with our spatial resolution, it would be quite difficult to interrogate a specific subsection of the PAG. Okay. This should be a movie. Let's see if it. All right. Cannot play media. Well, my apologies about that, but this should be a movie. A third and final challenge is posed by the physiological noise. So the brainstem is surrounded by spinal fluid and blood vessels. So this is the fourth ventricle, and this is a major blood vessel, and therefore more prone to physiological noise, which can corrupt the fMRI signal, especially in the dorsal aspect, where many nuclei are located. Therefore, particular care is needed in the analysis stage of brainstem fMRI data, with specific steps that can be performed in order to reduce the influence of physiological noise. A possible solution is given by ultra-high-field imaging, which provides enhanced spatiotemporal resolution and signal-to-noise ratio, thus allowing for smaller voxels. In this example, the difference can be appreciated, especially for the functional MRI data, where substantia nigra and red nuclei are distinctly visible in the 7T data here, as opposed to the 3T data in the below. An additional challenge comes from physiology, which tells us that vagus nerve activity and hence NTS response, so the primary afferent nucleus that we were targeting with TAVNS, are both synchronous with respiration. So NTS is a key node in respiratory sinus arrhythmia. That is the mechanism through which the heart rate increases during inhalation and decreases during exhalation, due to regulation of the efferent vagal tone via NTS and nucleus ambiguus. We also know that NTS receives sensory afferents from aortic baroreceptors and lung stretch receptors during inhalation, concurrently with top-down influence from the ventral respiratory group in the medulla, resulting in NTS inhibition during inhalation and facilitation during exhalation. All these factors have to be considered when using fMRI to measure NTS response, so to take into account its physiological oscillations. However, this physiological mechanism can also constitute an advantage in our case. We can then introduce the respiratory phase as a stimulation parameter and hypothesize that given that NTS activity is facilitated during exhalation, if we deliver TAVNS during exhalation, we can increase NTS activation compared to TAVNS delivered during inhalation. This also gives us the advantage of avoiding continuous stimulation, which could lead to possible habituation of NTS and brain response in general. We call this paradigm respiratory gated auricular vagal nerve afferent stimulation, which I will refer to as RAVANS from now on. So in the picture, you can see a diagram of the stimulation setup where respiration is measured from the subject and used to gate stimulation in real time. In our first study, we combined ultra-high-field fMRI, so using a 77 Tesla scanner, with brainstem-specific data collection and analysis methods to compare brainstem responses to exhalation and inhalation-gated RAVANS, which I will call E-RAVANS and I-RAVANS for simplicity. We enrolled 16 healthy subjects and used customized MRI-compatible electrodes to deliver stimulation to the Simba concha of the left ear. Subjects underwent separate 8-minute fMRI scans, during which they experienced either E-RAVANS or I-RAVANS in a counterbalance order. Additional scans included an active control, where the electrodes were placed on the left earlobe, innervated by the greater auricular nerve, and a zero current scan to control for respiratory influence on NTS response. The order of the 4 conditions was randomized across subjects with a similar number of stimulation events, interstimulus interval, stimulation current, and perceived intensity, as shown on the right. fMRI data were collected with a partial brain coverage centered on the brainstem. So when looking at target nuclei response, E-RAVANS showed robust activation of ipsilateral NTS, significantly higher compared to I-RAVANS. As for the active control, the response was more consistent with ipsilateral spinal trigeminal nucleus compared to E-RAVANS. This is a reminder of the importance of carefully choosing controls in TAVNS studies, especially when considering them as inactive. So we also compare the fMRI response to different conditions in 2 serotonergic source nuclei, the dorsal and the median raphe, and in a bilateral noradrenergic source nucleus, locus ceruleus. We found significantly greater response during E-RAVANS in both dorsal and median raphe, as well as in contralateral LC. And finally, we used electrocardiographic data, ECG, collected during the scan to estimate time-varying heart rate variability indices and looked at the evoked response of the high-frequency spectral component, known to be related to cardiovagal modulation. We found that E-RAVANS enhances parasympathetic output as measured by this index, but this is not found during I-RAVANS. At this point, we decided to optimize a second stimulation parameter, and we looked at the effect of stimulation frequency on brainstem response. Unfortunately, ultra-high-field MRI was not available for this study, so we doubled the sample size and enrolled 30 healthy controls and used a 3 Tesla scanner. This time, subjects underwent 4 E-RAVANS active scans, each at a different frequency, plus the zero current stimulation scan to correct for respiration effect. The frequencies considered in the study were 2, 10, 25, and 100 Hertz, and were delivered in random order across subjects. The stimulus amplitude, that is the current, was calibrated in order to match the sensation across frequencies and subjects. Therefore, lower current values were used for higher frequencies, as shown in the upper left graph. In this study, we also evaluated whether E-RAVANS had any effect on respiratory depth or frequency, and we did not find any differences compared to the zero current condition. When looking at target nuclei response for the 4 stimulation frequencies, we found that 100 Hz evoked the highest engagement in ipsilateral medulla, with a cluster encompassing NTS, spinal trigeminal nucleus, and dorsal motor nucleus of the vagus. Obviously, the resolution here is not as good as using ultra-high-field, so we had to be a little bit more careful in correctly labeling these nuclei. The second highest response was found for 2 Hz stimulation, suggesting a nonlinear dose-response curve. A subsequent source nuclei analysis, very similar to the one that we performed in our first study, also confirmed consistently greater activations in response to 100 Hz E-RAVANS, with significant responses in dorsal raphe and ipsilateral locus ceruleus for 2 Hz as well. Interestingly, a positive correlation was found between activations in these 2 nuclei at 2 and 100 Hz, meaning that subjects with a higher response at 100 Hz also exhibited a higher response at 2 Hz. So now that we have chosen 2 parameters that have shown to enhance the response of target nuclei in the brainstem and actually engage them, we can explore a possible application of RAVANS. While our lab has shown acute effects of RAVANS in chronic pain populations and in major depression, in this talk, I will focus on gastrointestinal applications, specifically for modulation of gastric function. The vagus nerve is known to play an important role in both sensory and motor aspects of gastric physiology, modulating gastric accommodation, that is the ability of the stomach to expand in response to food ingestion, and gastric motility, that is the ability to generate contractions for gastric emptying. Therefore, modulating vagal efferents with RAVANS may allow to affect gastric function when this is impaired, such as in gastroparesis or functional dyspepsia. Moreover, our frequency modulation study showed that RAVANS delivered at 100 Hz is able to engage the dorsal motor nucleus of the vagus, which is the main motor nucleus sending vagal efferents to the stomach. So the question is, how can we evaluate the gastric response to RAVANS? In order to answer this question, we need to know what gastric outcomes we want to measure and how. To evaluate gastric accommodation, emptying, and motility simultaneously, we need a test that provides volumetric assessments continuously over time. One possible option is given once again by MRI that has been proposed as a reliable, non-invasive method for detailed evaluation of gastric emptying and motility, without the disadvantages of ionizing radiation and the use of intragastric catheters, which are very common in the GI world, unfortunately. These main advantages provided by MRI, however, have been so far counterbalanced by as many drawbacks, thus limiting its widespread use. Most abdominal MRI sequences require breath holding in order to avoid image corruption by respiratory motion. Thus, volumetric images covering the entire stomach are usually limited to a single time point. For motility assessments, where images over time are needed, the field of view is generally limited to a single slice, thus losing the volumetric information. And finally, gadolinium is often used as a contrast agent, with contraindications in vulnerable populations such as renal insufficiency, pediatrics, and pregnancy. To overcome these limitations, we have proposed a free-breathing, 4-dimensional MRI sequence using a food-based contrast that will be used to assess the effects of RAVANS on gastric functions. So in our ongoing study, 15 healthy controls and 12 functional dyspepsia patients were enrolled for 2 MRI visits. Each subject was asked to fast overnight prior to each MRI visit, and then received either sham, meaning zero current, or active RAVANS for the entire duration of the visit. At the beginning of the session, subjects consumed a fixed dose of a pineapple-based, semi-solid, high-caloric contrast meal. It's delicious, I make it, it's great. Following the food ingestion, they were placed in a 3T MRI scanner, and continuous abdominal volumetric images were collected for 5 minutes at 3 post-meal time points, namely after 15, 65, and 80 minutes following food ingestion. The MRI sequence used did not require the subject to hold their breath and provided full coverage of the stomach in every 7 seconds. A mosaic movie of unprocessed abdominal images is shown for a representative subject here on the left. Following data collection, the gastric volume is segmented within the images via a semi-automated procedure. Briefly, the manual segmentation of the first volume is then propagated on subsequent time points using a nonlinear volumetric warping. Our first paper came out just last month, and we demonstrated to be able to measure gastric emptying, separating meal and air content in the stomach, and also look at gastric motility by identifying the propagation of peristaltic waves along the gastric walls and extracting parameters such as peristaltic frequency, enteral occlusion, and velocity of wave propagation. We are currently testing this paradigm in patients with functional dyspepsia, as I mentioned before. Functional dyspepsia is a disorder of gut-brain interaction characterized by numerous symptoms such as nausea, early satiation, abdominal discomfort and pain, and bloating, and there are many symptoms. Preliminary results show that active RAVANS had a significant effect on gastric volumes and meal emptying. Specifically, total and meal volumes were significantly larger at T0, so immediately after the ingestion of the meal, even though subjects consumed a comparable amount of food in both visits. This finding might suggest increased accommodation, meaning that the stomach is able to expand more easily in response to food. In response to food during active RAVANS. Moreover, gastric meal emptying measured at 65 and 80 minutes post-meal were significantly higher during active RAVANS compared to sham, as shown here on the right. And interestingly, no effects of RAVANS was found in the healthy control groups. So this is just a small part of our complete design. In the full protocol, we alternate gastric MRI with brain MRI, fMRI, to look at the so-called gut-brain axis. We aim to use a functional localization of the NTS that we obtained with our first study at Ultrahigh-field, which is, as I said before, a major node for visceral afferents carried by the vagus nerve, and then link measures of functional properties of the brain and the stomach, and ultimately evaluate the brain-gut axis in health and disease. In conclusion, in the first part of the talk, we have shown that brainstem neuroimaging using fMRI allows to optimize RAVANS target engagement. And I have then described a comprehensive tool based on magnetic resonance imaging for integrated non-invasive brain-gut assessments of RAVANS effects for GI applications. And finally, we have proposed an application of RAVANS in GI patient populations and showed promising results, hopefully, for this intervention in modulating gastric function in FD patients. And I would like to conclude by thanking my mentor, Vitaly Napadov, and all my collaborators and lab members, as well as funding sources. And of course, thank you for your attention.
Clap, clap, clap. Hey, nice job, Roberta. Thank you so much for joining us. And I think people can probably tell why I'm really excited to work with you. You know, we've got a grant proposal out now, and hopefully we'll hear back in some good news. So one of the questions that I wanted to ask was related to the RAVANS stimulation, where the contrast that you've shown is inhalation versus exhalation, or sham or placebo, I guess, versus exhalation. And that's a within subjects contrast. And I'm curious if you think that you'd be able to pick up fMRI differences showing a difference in deltas between a patient group and a control group. So for example, could you show an inhale exhale difference in a patient group like in ME/CFS or whatever it is, you know, some of the patients that we think may have a problem in this circuitry versus controls?
That's possible, but I would be more interested in showing a difference between controls and any patient population within the exhalation stimulation. So the good one, because we have found that the gastric response is only there for patients and not for healthy controls. So perhaps also the brainstem circuitry is more engaged in patients because there is a deficit. You know, if you start from a lower point, then the effect size is bigger. So there might be a difference in that.
That makes sense. And if I may, can I ask another question? So
Yeah.
one of the things that, you know, with vagus nerve stimulation and the auricular transcutaneous vagus nerve stimulation is that there's some individualized responses. You know, for some folks, it seems like, you know, whatever 1 hertz versus 25 hertz. And, you know, that and it seems to be almost like certain settings are inhibitory for some, excitatory for others, and that sort of thing. What do you know about the individual variance in the sort of settings, and how much does that affect experimental design?
So in general, and you will find, if you just look a little bit in the literature, you will find plenty of reviews highlighting this issue. There is a lot of variability in the field, starting from the location of the stimulation, even the anatomy of the nerve afferents in the skin, you know, individual anatomy of the ear and nerve endings. So it's very hard to standardize and to compare different studies. That is a known issue. Also, the choice of the sham or the control is very highly debated, because a lot of groups choose the earlobe, and we tested that as well. But the earlobe is not part of the ear. You know, it has some innervation, so you will see a response. And if you don't have the resolution to discriminate that response from the one you're looking at, they would basically overlap. So you would not see any difference. We just saw it because we used ultra-high-field. So yeah, it's very hard.
Well, I'm looking forward to working all this stuff out with you. So thanks very much for joining.
Thank you.
All right, cheers.